Disruptive Technologies, Section 6, Lesson 1, Next Generation Genomics. The science of genomics is at the beginning of a new era of innovation. The rapidly increasing cost of gene sequencing is making huge amounts of genetic data available, and the full power of information technology is being applied to vastly speed up the process of analyzing this data to discover how genes determine, treat or mutate or cause diseases. Armed with this information, scientists and companies are developing new techniques to directly write DNA and insert it into cells, building custom organisms and developing new drugs to treat cancer and other diseases. Every cell in our body contains a copy of our genome. Over 20,000 genomes build 3 billion models of DNA. DNA consists of two strings twisted into double helix and held together by simple pairing rules. A pairs with T and G pairs with C. To understand the impact, researchers using DNA sequencing technique. Researchers have identified thousands of genes and affect risk of diseases. To understand how our genes work, researchers needed a way to control sequencing. The potential economic impact of next-generation gene sequencing in the application that we have sized in healthcare, agriculture, and the production of substances such as biofuels could be around $1.6 trillion by year 2025. About 80% of this potential value would be realizing through extending and enhancing lives through faster disease detection, precise diagnosis, new drugs, and more tailored disease treatments. Agriculture analyzing plant genomes could lead to more advanced genetically modified crops and further optimize the process of farming by tailoring growing conditions and farming processes to accede genetic characteristics. Furthermore, it may be possible to create high-value substances such as biofuels by modifying simple organisms such as Escherichia coli bacteria. Easy access to gene sequencing machines could not only put power genetic technology in the hands of researchers and physicians, but could also create a global community of co-creators and that might advance biotechnology in unforeseeable ways as hobbyists propel the microcomputer revolution. Next-generation genomics can be described as the combination of next-generation sequencing technologies, big data analytics, and technologies with the ability to modify organisms, which include both recombinant techniques and DNA synthesis. Next-generation sequencing represents newer, lower-cost methods for sequencing or decoding DNA. Encompasses the second and third generation sequencing systems now coming into widespread use, both of which can sequence many different parts of genome in parallel. This advance in DNA sequencing speed promises to accelerate the process of biological discovery. Historically, biological research has relied largely on hypothesis-driven trial-and-error testing. This approach is very time-consuming and difficult, so scientists' understanding of which genes drive specific outcomes remains very limited. Advanced genomics will also facilitate advances in agriculture. Farmers might be better able to optimize soil types, watering schedules, crop rotations, and other growing conditions based on more complete understanding of crop genomes. It may also be possible to produce genetically modified crops that can grow in locations where soil conditions and access to water cannot be easily improved. Crops that thrive in colder, drier climates, or crops that generate a larger portion of their weight as food. The next generation genomics technology could be used to modify DNA of common organisms to produce valuable substances. Using synthetic biology or even standard well-established recombinant techniques, the metabolic system of certain organisms can be modified to produce specific substances, potentially including fuels, pharmaceuticals, and chemicals for cosmetics.